Segregation biology classes. Today our topic is about the cell cycle. So the first question is, what is cell cycle? Cell cycle is actually a biomolecular process by which a single cell divides either two or more cells having maybe same or different type of nuclear material. So the cell cycle divided into two major categories. One, here we can see that mitosis, another one is the meiosis. In the mitotic cell cycle, the mother cell divide into two daughter cells. Here the cell number, one cell is divided into two cells, where the cell number are 2 and their genetic material is equal with the mother cell. Here we can see that the 2n number of chromosome, that is the 46 in case of our 46 number of chromosome, are duplicated, and the new daughter cells each have the same number of chromosome, that is 2n or diploid number or 46 number of chromosome. This is the mitosis, where the mother cell divided into two daughter cells having the same amount of genetic material. In the other hand, the meiosis is also cell cycle where the mother cell divide, but here they divided into 4 different type of daughter cells, each having the half of the genetic material from the mother cells. So here we can see that the mother cell have 2n number of chromosome, while the daughter cell have the single n number or haploid number of chromosome. So in both cases, whatever in the mitosis and meiosis, there must be an intermediate stage between the mother cell and daughter cell, where the genetical chromosome number may be 4n. So here in the mitosis we can see that the 2n number of chromosome is duplicated into 4n number of chromosome and then split into 2n and 2n. Where in the meiotic cell cycle, also called meiosis cell cycle, where the mother cell 2n number of chromosome is duplicated into 4n number of chromosome, and then it is split into single n 4 type of cell. Now the question is, which type of cell is undergo through the mitotic cell division or mitosis cell division, and which kind of cells is undergo through the meiotic or meiosis cell division? The answer is very simple. All the cells, all the body cells, including stem cells, somatic cells, undergo through the mitotic cell division, except your germ cells, that is sperm and ovum. Only the sperm and ovum present in the testis and ovary are undergo through the meiotic cell cycle. It is so necessary because to form a zygote, that is the 2n number of chromosome, this is called a zygote. It's actually made due to fusion of an n from an egg and n number of chromosome from a sperm. So it is very necessary to produce the half cell in the sperm and ovum. Half cell means the half genetical nuclear material containing cells to produce the zygote, which is become a 2n number. If the meiotic cell cycle will divide as like a mitotic cell cycle, and each cell having the 2n number of nucleus, then the maybe assume that if sperm go through the meiotic cell produced by the meiotic cell division, and sperm containing the 2n number of chromosome and egg also containing the 2n number of chromosome, then zygote will be in 2n number of chromosome. It is very necessary to receive half genetic material from the egg and half genetic material from the sperm. So egg and sperm, in each cases it is very necessary for them to receive half genetic material cell from their mother cell by the process of meiotic cell division. But today our lecture is focused on the mitotic cell division. So we understand what is the mitosis and meiosis, then mitotic cell division. As I told you earlier that the mitotic cell division or mitosis is a cell division where the mother cell is divided into two daughter cells where the cellular chromosome number is equal between the mother cell and daughter cells. So that there are, if we assume that the cell cycle, it is in a cyclic process like this, it is a cyclic process. This cyclic process are divided into two major categories. Whatever it is a mitosis or meiosis, a cell cycle is divided into major two categories. This half portion is called the I phase or interphase, and this is half portion is called the M phase. Now M stands for two cases. If the M phase may be mitosis or M phase may be meiosis, but the I phase or interphase is common in both in mitotic cell division and meiotic cell division. But after the interphase, if the cell undergoes through the meiotic cell division, it is known as the meiosis, and if cell is going for the mitotic cell division, it is known as the mitosis simply. So I phase or interphase, this interphase is divided into three major sub stages. One is known as the G1, another is known as the S, and the G2. Now G1 is also called G1 phase or growth phase, as the G2 is also called the G2 phase or growth phase, where the S stand for the synthesis. Now what happened in the G1 phase? G1 phase is the very important, it's an extremely important phase where cell growth take place. That means all the necessary proteins are developed in the G1 phase and cell is preparing for the S phase. It is the role of the G1 phase. So what happened actually? Two mother cell wanted to divide into two daughter cell having the same chromosome number. So it is very necessary to first double the volume of the chromosome, that is 2n to 4n. Then the 4n can be split into 2n to n, the simple mathematics. So the S phase is responsible to duplicate its chromosome into the double volume. That is that in the G1 phase, if we study the chromosome number, we can, not number, the chromosome volume. The chromosome number, it is a very important point that chromosome number will never ever change in the cell cycle, especially in the interphase. Only the chromosome volume will change. The chromosome volume or the chromatid volume in the G1 phase, we can see the 2n number, where in the end of the S phase it is become 4n number. So it is the major phase where the chromosome number get doubled. How? The chromosome is actually, this is a chromosome, this is the look like a structure, this is the chromosome. Now chromosome is actually made up by the DNA molecule. The chromosome is actually made up by the DNA molecule. So it is very important to replicate the DNA. A single DNA will replicate it into the two DNA molecule, by which from here we can having the two sister chromatid. So that means it is made up by the DNA, and DNA is getting in doubled in volume, so the chromosome will become the doubled in volume. So this is all about the S phase. Now after the S phase, it is very important to check the DNA for any misincorporated nucleotide in the DNA. So to check the newly formed the DNA is checked in the G2 phase. So that is the important of the G1 phase, S phase and interphase or G2 phase, that together known as the interphase. But this, as this phase are containing the growth phase, it is very important, it is very lengthy process. However, in cell to cell, this cycle is being actually time is different in manner. But however, the S phase is containing 8 hours, where the G1 phase itself containing 11 hours, where G2 is containing the 4 hours. While the only nuclear segregation take place in the M phase. Only the nuclear segregation is taking place in the M phase, which is last only for 1 hour. Okay. So now we are talking about what is the embryonic cell cycle. What is the embryonic cell cycle? Now this zygote, as this zygote is formed, this is undergone through the rapid mitotic biphasic cell division. It is known as the rapid mitotic biphasic cell division. That means as the zygote formed by the fusion of sperm and ovum, it undergoes through the mitotic cell division, but this mitotic cell division is very rapid. That means the time consumption is very low during the zygotic cell division compared to the normal mitotic cell division. And biphasic. Biphasic means that this kind of cell division do not consist, I repeat, do not consist G1 and G2 phase. There is an interphase and there is an M phase, but interphase only containing the S phase for the DNA replication phase and followed by the M phase, mitotic cell division. So during this is known as the blastomer stage. Okay, this is known as the blastomer stage. During this condition, the cell is rapidly divided. S phase followed by M phase, followed by S phase, followed by M phase. This is going on. But later, during the mid-blastular transition, G2 is incorporated and then the G1 became incorporated and the normal cell cycle became start. So this is all about the overall idea about the interphase. But we will later discuss on that mitotic is further. Mitosis is known as the nucleus segregation stage. Mitotic and meiotic both are known as the nuclear material segregation stages, which is further divided into prophase, prometaphase, metaphase, anaphase, telophase, and finally the cytokinesis, where the cytoplasm of the cell is divided equally in the both cells. So friends, as we discussed, the mitotic cell cycle is divided into two major categories. One is the interphase and another is the M phase. But what happened after the M phase? Because up to now we understand that during the interphase, the cell preparing for the replication in the G1 phase, this nuclear material get doubled in the S phase, and the G2 phase, the nuclear material is checked for the any mutation. Think about, there are a lot of proteins in the G2 phase that are responsible to correct if there is a misincorporation or DNA mutation. It is very simple. Think about it, that some protein are mutated or non-functional during the G2 phase. So if there is any DNA replication error happen, it will be not checked by the G2 phase, and that will lead followed by the cancer. We know that the cancer is responsible for the mutation, but mutation is a spontaneous process. In every cell in our body that are undergoing through that cell division are mutated sometimes, but the mutation is checked in the G2 phase. But in the cancer patient, what happened, the mutation, the error in mutation are unable to check by the G2 phase, and that will lead further and produce the cancer. So what happened when the new mitotic cell division is occurred? After the mitosis, what happened? There is a G0 phase after the cytokinesis. G0 phase is a very important term. The G0 phase also called growth zero or quiescent phase or G0 phase. So many terms: G0 phase, quiescent phase or G0 phase. What happened? In this phase, the think about it, that the daughter cell are produced it is became in a G0 phase, that means this cell will not divide. It is perform its normal housekeeping function. For an example, think about it. If this cell is the islets of the Langerhans beta cells, and this cell is responsible to production of the insulin. So the insulin production is the housekeeping function of the beta cell of the islets of the Langerhans of the pancreas. So it will be just perform its housekeeping function. But sometimes cells are divided, and some cells do not get divided after they become differentiated into a particular cells. So this is depend upon the G0 phase. If after the G0 phase, if a cell is again enter into the G1 phase, then it will be divide further a cell division. But if they remain in the G0 phase, they will not divide. So according to that basis, the mitotic cell division is further divided into three types that are developed in different type of cell. First, we know as the cell of our body, that is somatic cells, not the germ cells, because the germ cells for meiotic cell division. We will not consider such cell in this lecture. We are considering the somatic cells and the stem cells in this lecture. And in our body, in human body, we are made up by the eukaryotic cells. So in our body, we have a three type of cells. It is very beautiful information that the first is a labile cell. Labile cell. The cell, those are never enter the G0 phase. Some of our body cells are continuously divide, such as the hematopoietic stem cell. Hematopoietic stem cells. They are labile cells, because we know that blood cells such as RBC, WBC are continuously produced in our body. But these cells produced from, these are the mother cells of the RBC and etc. blood cells. So they are continuously dividing. They will never stay in the G0 phase. So this is the labile cells. Another important beautiful example is the skin cell. We know that there is always production of the skin cell. Okay, skin cell, that is the labile cell. That is the mean when we are in a hot weather or hot climate, our skin cells become damaged. But if we protect ourselves, our damaged skin cells are eliminated by the fresh skin cells. Another important is the gastrointestinal cells, such as we know that in the gastrointestinal system the absorption is taking place by the villi. These are the intestinal cells. So these are continuously produced from its mother stem cells. These are the labile cells. Another part is the occasionally dividable cell. Occasionally dividable cell. What happened in the occasionally dividable cell? That means after the differentiation into a particular cells, they will never divide until it is very necessary to divide. For an example, liver cell will never divide, and after the differentiation from the stem cell, they remain in the G0 phase and they perform their housekeeping function. The liver cells is also known as the liver hepatocytes. But experimentally, if you cut a very tiny volume of liver, you can see that the growth is happened and liver is again produced in a fully well differentiated manner. So liver cell, lung cells such as lung alveoli, these cells do not undergo cell division. They remain in the G0 phase. But if in the and also the cardiac cell, the heart muscle cells, it is also important. Cardiac cell, heart, they are never ever divide. But if it is necessary, they will sometime try to divide it. But heart is not so important. It is actually not divided in that way. But lung and liver will be definitely occasionally divided. And the number 3 is that the cells, they were never divide after the differentiation process. The beautiful example is neurons or nerve cells. After the fully formation of the and differentiation of the neurons, they will entire their life remain in the G0 phase, where they will never uh, divide and produce the new fresh neurons. So what happened? In a very good example, if I tell you, if your skin is cut, blood loss happen, but it will take 3 days, 4 days, 1 week, but the skin become damage, will become repair, because it is a labile cell, so the cut will be repaired because it is a undergo through the continuous cell division. What happened in the occasionally dividable cell, if a jaundice may happen or any hepatitis happen and some of the hepatocyte cells became damaged, so normally liver cells do not get divided, but in that case the liver divided, cells are divided, and fully good liver became another time formed. But what happened, we know that in case of the cerebral attack, the patient are suffering with the paralysis are never overcome from that disease, or in Alzheimer or in schizophrenia, these diseases are very extremely impossible to cure because the damage of the neurons, because neural cell is never divided, so there is no way that your damaged neurons will be replaced by your fresh neurons. That will be never happen, so that the neural damage will never repair, and the people with the neural damage must suffer with this damage until their last breath. So this is all about the cell cycle introductory phase, about the mitosis, meiosis, type of cell cycle, interphase, M phase, and embryonic cell division, and all about the labile cell, occasionally cells, and never dividable cells.